Yeah, it's working. So Pierre-Olivier Schmit is the business development manager for the proteomics team at Bruker. So he's here to tell us the amazing things about the timsTOF.
Yeah, thanks for the introduction, Simone. I really apologize for not being able to be here today. It's a pity. Fortunately, my colleague Martina is there, and I hope you enjoy a very nice meeting. It sounds great, actually. So now I have the challenge to keep you awake after that heavy lunch of yours. And I will just give you an overview of how another vendor can see how we can develop things to facilitate routine single-cell proteomics analysis. And I really want to stress on the routine part here to try to make it more a journey from the exceptional results to something you can get on a day-to-day basis to really use it for science. So the conference has also already started, and you've described a lot of things. So in the agenda, I will just skip the two first parts because I think the point has been very clearly made of the interest of single-cell approaches towards bulk approaches, and on why proteomics is a nice complementation to genomics and transcriptomics approaches. So I will rather focus on what makes the specificity of the technological specificity of the approach we are proposing, and then try to replace it under the view of what is required for single-cell analysis. Describe a bit how we at the LCMS level were trying to chase for more sensitivity, and how we do, or we have tried to integrate that into a complete solution, because it's not only about LCMS as it has already been described. And with that, I think we will cover the time allowed for this presentation. So, starting with the beginning, I'm sorry, I will use awful acronyms like trapped ion mobility spectrometry for TIMS and PASEF for parallel accumulation serial fragmentation, which basically are the specificity of the family of instruments we are using for these applications, which are the timsTOF family. And the approach, we name it 4D proteomics, and I will try to explain what we mean by this. It's not only marketing, I can assure you. So the timsTOF pros are systems which are based on a cutoff basis together in which dual trapped ion mobility cells have been mounted. So practically it looks like this with a 90, I will use my pointer here. So we have also taken a special effort into designing the system for robustness, whatever the application is for. And that's why you have a first 90 degrees angle, which is hardware free here, to allow all the neutrals to get through. And the specificity of the system is that it basically combines different separation technologies like HPLC, like all LCMS MS, but also very specific trapped ion mobility device, which is in front of the QTOF system. So I won't go into every specificity, but the things to notice is that the separation timescale of each separation fits very well together with the next one, which means that in an HPLC peak, there is plenty of space to make several TIMS experiments, so to sample properly the HPLC peak and get nice quantitation results. And in the same way, in each peaks eluted from the TIMS experiment, it can be coached on the flight by the quadrupole, which can operate up to 350 Hertz. And in each of these very narrow peaks, they can be sampled by the very quick time of flight system. So at every stage, the sampling is correct and ensures for good outcome and quantitation. But the magic here relies in that purple area, the trapped ion mobility separation system. So to make it short on how it does work, if you look on the right, this is very schematic. We have two trapped ion mobility cell. in which we have counter affecting forces from the air entering the mass spec, which pushes the ions into the mass spec with a strength which is increasing with the hydrodynamical radius, the collisional cross-section of the ions. And on the other hand, an electrostatic field of increasing strength, which slows them down. So that means that we can stop the ions at one position in the funnel, and we can then release them by in an extremely precise way by lowering this electrostatic field value. And when we do that, when we do this evolution in the second cell, the first one is filling up to avoid losing any ions. So the effect is not only a separation based on the collisional cross-section, it's also time and space concentration, because all those ions collected, for instance, for 100 milliseconds are then released as 2 to 4 milliseconds with peak. So it's very dense packets of ions which can be caught on the flight by the quadrupole. That's what we call the parallel accumulation and serial fragmentation. And doing so, we can not only have an extra dimension of separation, we can also increase at the same time the sensitivity and the speed without losing the selectivity. And that makes the specificity of those instruments. On top of this, for every eluted peptide, we can determine a collisional cross-section value, which we will also use extensively for identification and characterization processes. And that's basically what supports what we call 4D proteomics. So 4D stands for the use of the different dimensions, like the retention time, the M/Z value of the precursor ions, its MSMS fragmentation pattern, and the collisional cross-section value, which is associated with the parent ion. So doing so, we benefit from the extra separation in the ion mobility dimension, as you can see at the top. So here you have an LC chromatogram, and on one peak, we clearly spread the complexity of the MS scan along not only the M/Z axis, but also along the mobility axis. On top of that separation, which, for instance, allows also for the separation of some isobaric compounds, and that clearly helps in terms of epi-proteomics approach. That means, for instance, when you want to, you can separate two coeluted position isomers of phosphopeptides where the phosphorylation is located at different locations. That's one example. But we can also use the numerical value of the collisional cross-section, and we actually do it extensively to improve the performance of the identification process by comparing predicted collisional cross-section value to measured collisional cross-section value and introducing that into an extra scoring algorithm, which allows to clearly increase the sensitivity of the search itself. So that's what we mean when we are speaking of 4D proteomics approach. So it sounds already well suited by its speed and sensitivity to single cell approaches. But if you take a step back, so of course, there are many hurdles linked with single cell analysis, and we had an extensive description of those already. So I'm skipping the cell culture and preparation part, which is out of the scope of my presentation. But if you just take the analytical part of it, of course, we have to face the sample preparation issues with a very low amount, which are concerned. Every touch, I would say, of the sample with the plastics is a loss, and we have to reduce that as much as possible using as low volume as possible. We have to magnify as much as possible the sensitivity of the acquisition process, not only on the mass spec, but at every stage, LC, mass spec, processing. And as already mentioned, we are dealing with biological sample, human sample, with all their variability, both at the cell level or the individuals levels. So that means that to get decent statistical power, we also need a number of analyses. So that means throughput is of interest, as already described, and robustness is also of interest because any downtime of the instruments will negatively influence that throughput. And that's where the processing pipeline, on top of delivering identification and quantification results, is also important, as it can influence the throughput negatively if, for instance, all the identification and quantification processes are taking too long. So these are a large set of possible hurdles, and I will now show how we are trying to address those. First, by getting on the high sensitivity acquisition process side. And as you will see, that is also linked with the high throughput and robustness, because we are basically answering those three simultaneously. So we've already had a nice view of possible solutions to get along with very low amounts. And traditionally, when it came to LCMS, affinity-based enrichment, starting with IPs in the early days, has already been a very nice way to enrich a sample in a specific way. More recently, carrier-based enrichment, so TMT-based approaches have been developed for single cell, and they have been quite successful actually, and they are highly represented in the literature. And nevertheless, label-free approaches are also very desirable for a couple of reasons, which we will see just after. So how do we deal with these three different approaches? When it comes to affinity-based enrichments, so there are, as for every method, several pros and cons. The sensitivity increase cannot be debated for sure. It's highly effective, and a nice thing, it can be also linked with high spatial resolution, so it can be used for imaging approaches. The costs are main cons are linked to the costs of this approach, especially if you want to multiplex, because you will increase the costs as much as you multiplex basically. And you are also restricting your analysis to what you are enriching. So the specificity of your analysis also depends on the specificity of either your antibody or aptamer, and you cannot get beyond that. Nevertheless, it's quite helpful. And for instance, as for us, we are using it especially with the imaging approaches, which can be a prelude to the single cell approach by allowing to, for instance, select a subset of cells that will be targeted for laser capture microdissection and further, I would say, real single cell analysis. The scope approach has been described extensively already, and it's quite appealing. It's a very clever use of the tandem mass tag approach, which has proven efficiency. Nevertheless, it's of course not without drawbacks. Normalization efforts, for instance, are a bit higher when you exceed 1-plex, because you need to reserve a channel for normalization. That's doable, of course. You cannot overload the carrier unless you want to sacrifice a bit of accuracy. But the two main points are probably that it's still a data-dependent analysis-based acquisition. So that works very nicely, but that has a negative influence on the throughput and on data completeness, which means basically if you want to reach a very high data completeness, you need to have a bit longer gradients to make sure you reduce the stochastic effect of ion selection. And of course, if you use it at a very large scale, it can be costly. So nevertheless, our system, as most mass specs, do support this approach with the limitations which are linked to the TOF analyzer. So we do not do more than 9-plex. However, it does work pretty well. Here is, for example, a communication from the Ben Osborne group. And basically, they compensate the lower number of plex by a faster analysis, which is allowed by the speed of the system. So basically, it's equivalent at least to what they did on the same sample with the scope approach, with shorter gradients and a bit higher number of cells linked to sensitivity of the system here. Nevertheless, and for some of those reasons, the label-free approach seems to be appealing. Why? It can be very easily applied to a very large number of cells, and that's, of course, cost-wise, extremely important for some labs. The most interesting probably is that it's compatible with DIA, and the DIA approach right now are unbeatable when it comes in terms of throughput for a single sample, non-multiplexed. for their capacity to deliver deep proteomic information from a given sample, and especially, oh, I wrote 2 times throughput. I was meaning, sorry for that. I was meaning data completeness. In a DIA approach, the difference between the number of proteins identified and quantified over a large number of samples is much lower than it is on the DIA approach. And that means basically you can basically quantify a similar number of proteins with much shorter gradients when you do with DDA. The cons are you cannot increase the sensitivity this way. So you have to fully rely on the sensitivity of your analytical pipeline. And that's why it's desirable to increase that. So that's what we tried to do already a year ago when we launched a specific system which we call the timsTOF SCP, standing for single cell proteomics. So that's a redesigned front end of the instrument. All the back end, which you see here, is common to, that's basically the former image I've shown you. And you see that instead of the capillary here, we have a front end with a much larger capillary, which allows to let more ions enter. But if you let more ion enter, we also increase the risk of contaminating the instrument. So that's why we had to redesign this front end, adding a further pumping stage to ensure better absorption and an extra 90-degree angle to maintain robustness here. And the outcome is a system that is basically, that preserves all the capacities of the former ones, but is basically 4 to 5 times more sensitive. So we've, of course, benchmarked it. And these were the first outcomes. So we first, of course, tested it with standard commercially available digests, and as you can see, with gradients as short as 15 minutes, we can already identify close to 2,000 protein group IDs from a 0.2 nanogram injections. So it looks nice. All of that, of course, is obtained with with DIA or DIA path analysis and an in-house search engine. I will describe this a bit more, which is based on a Bruker rewritten version of the DIA NN software, which also operates in real time. But that's for later. The system as it was has been beta tested, I would say, in the group of Matthias Mann, and that has already raised some nice results and a publication. It has been initially tested with the first EvoSep1 systems that were capable of operating with a Whisper NanoFlow setup. So it was a routine setup in terms of robustness, and the outcome have been pretty good, actually. So they made a very nice study where they stopped cells at different stages of their cell cycle, compared that to transcriptomic results, and yielded some very interesting results. I especially want to focus on that graphics here, which really shows that it's possible to quantify the number of quantified proteins at a large stage. I think there were 420 proteins here quantified in that study, and that made it to a molecular system biology. I would already say that were the early stages, as both the LC system, the DIA method, and more than this, the sample processing, the data processing pipeline were not at the stage they are today. So if you want now to merge all of the progresses which have been made, I would say in the last month, because it's a very fast evolving area, I will now focus on our collaboration with Selenium when it comes to lossless sample preparation and tell a bit more about the processing pipeline. So as mentioned, and I think this was mentioned already in some other talks this morning, you can have a very nice Ferrari, that's not enough, and you might not be able to do everything you want with it. So having in the same parallel a very nice LC-MS system doesn't mean you will be successful in all analysis. So you have also to resolve all issues we have before and after this. And especially in terms of preparation, I especially like this picture because in terms of very sensitive sample preparation, you can have extremely skilled person in the lab that do better than any robotics, but it's some kind of wizardry. And if you lose that very experienced PI or postdoc, then you lose basically the capability. So it's kind of important to have something reproducible in terms of sample preparation. And at the other edge, it's also important to have very reliable sample processing systems. And we do here rely on the in-house parallel search engine in real time, which is a Pazar solution, which features DDA and DIA search algorithm. The DIA one being based on DIA-NN, which has been rewritten to better take into account the collisional cross-section value into the scoring. And one particularity of the Pazar here is that it's really searching in real time. So it does not introduce more downtime in terms of search processing. Coming back to the sample preparation, so as said, we are collaborating heavily with Selenium. So David made a very nice presentation already, and you will see some common results because we are basically working in common. As already mentioned almost a year ago, we made the first tests, and the samples have been prepared by Anjali in Lyon at Selenium, and frozen, shipped to Bremen, resuspended in a large volume by Christoph Crisp to put them in a microtator plate and then measured on our standard nano-LC IMaSS system with the DIA Pazar methods on one early version of the Pazar system. And even at that time, we've been pretty nicely surprised because we were already capable of identifying an excess of 1,400 protein group ID per single cell, with a nice increase while we were increasing the number of cells. And we continued to work on the different workflow optimization, and especially a huge work has been done at the Selenium level, where they have developed the label-free protein chips, which particularity is to enable to take it directly out of the Selenium system and to place it into the autosampler of the nanoELUT, therefore avoiding all those very risky steps of resuspension, dilution of the sample, where we basically lose a lot of the material. And while either while doing this or while loading directly from the Selenium, from the protein chip output to EvoSep tips to benefit from the robustness of the EvoSep system, the outcome now with both these approaches, so either the standard nano-LC or the EvoSep one, with this, while suppressing this step where we redilute, are even more impressive. From the same kind of sample, still prepared in Lyon, still frozen, shipped to Bremen, but this time not resuspended, then we can identify an excess now of 2,000 protein group ID from a single cell HeLa cell line separation. So very nice progresses, and we are quite happy with that. And progress, it's still ongoing. Still improving, I would say. As mentioned already, and we are not chasing about benchmarking example. The final purpose is to make science and to perform quantitative biology on these very low amounts. And here is another example of what we can get when we try to clearly understand what the system is doing. So my colleague Christoph Crisp here has basically loaded EvoTips with given amounts on the cell of HeLa cell line digest on the tips, and compared it to preparation of different cell amounts on the EvoSep one, and then it tracked the intensity for some specific proteins. So here I've chosen two of those, the 60S ribosomal protein L6 and the endoplastic reticulum chaperone BIP. And roughly, we estimate that in one HeLa cell we have something like 0.25 nanogram of protein, and that for that protein, it was nicely fitting in terms of intensities for one cell. And you can see that if you multiply the number of cells by 5, and you take that value from 0.25 to basically 1.25 here, we end up with this amount. So it fits well, and the same applies to an increase from 1 to 10 cells. So it looks like the quantitative answer is also reliable, and of course that is backed up by a lot of other experiments. I do underline this because we are really challenging the system here, not only due to the low number of copy of proteins and the low signal that we have, but also with the very short gradient we are using on still very complex sample. All data sets are quite crowded, and here the capacity of the system to spread the complexity along the mobility axis and the capacity of the post-processing system to make a full use of this extra dimension and take the collisional cross-section coding of the peptides into account to make the identification and quantification more selective is also accountable for the success of the experiment. So it's not a mere raw data processing, but yields to that. It's the final test of the complete pipeline, basically. And I guess that will be covered extensively by Nikolai Slavov, but I can't resist to not to present, I would say, to show the perspective here of Nicolas will probably describe that he's working on Plex DIA. This has also been mentioned earlier this morning, and I was mentioning the potential of DIA approaches in terms of throughput, and Nicolas is currently trying to push things further by multiplexing DIA, and we clearly see that as a very exciting perspective, further enabling to increase throughput without damaging the analysis depth and quantitation capabilities. So things are not over in terms of performance increase when it comes to single cell proteomics analysis. As some sort of wrap up and conclusions, so what you are trying to do with as a provider of solutions for you to use is to try to address those main hurdles. So it comes through collaborations when it comes to the sample preparation, with collaboration with Selenium, the development of proteochips and other protocols that they are working on. We still continue the developments of the timsTOF family of instruments, and the SCP is clearly designed to address those low sample amounts, both at the sensitivity level, but also in terms of throughput capacities, where it's backed up as well by fast LC system like the EvoSep one, and it's clearly helped by the DIA PASF approaches, and also the post-processing analysis that doesn't kill the throughput by inserting a low throughput identification workflow. The robustness is key here. Also, it comes also from every part of the solution, whatever it's the LC part, the captive spray source, which is also designed from robustness, and of course the mass spec itself, which is also designed for low maintenance. And again, the processing pipeline has also its importance, as it's basically the one transforming the data into useful knowledge. With that, I have, of course, a lot of person to thank, thanking you for your attention, thanking my Bremen colleague who generated all data generated in colleagues, our collaborator in Martin Street for the early data, EvoSep collaborators and Selenium collaborators for helping us setting the system or preparing the sample in Lyon. And of course, I thank you again for your attention.
Thank you, Pierre-Olivier. You got a big round of applause. I don't know if you can hear that.
No.
So a couple of quick things. So number one, I need a volunteer for the microphone. Thank you. And number two, just as a reminder for the speakers, for you and everyone else, I noticed that you had some slides with written confidential. If you don't want your presentation to be recorded and put online, let us know, or now or later by email, so you will not end up on YouTube.
No, it's not an issue. I mean, the confidential is automatically set up by PowerPoint.
Perfect.
I just forgot to remove it.
Okay, questions for Pierre-Olivier? Yeah, a question from Carl.
Hi, Pierre. It's great to see the big progress of Bruker in single-cell proteomics, and a wonderful presentation. One strategy was not quite clear for me, yes? Because you presented data with the nano-LO2, later with the EvoSep, and looks like the EvoSep outperforms the nano-LO2. Yes.
It's
if your data are correct.
yeah, it's a shortcut, Carl.
Don't tell me your data are not correct. Yeah.
The data are fully correct, and you're right. You have a sharp eye, actually. No, there are different things. The one I presented. So basically, to answer your questions, we roughly get the same level of performances nowadays with single cell preparations. The main differences is it's not the main differences. We get it with a slightly shorter gradient on the nano-elute than on the EvoSep, but shortly means 25 minutes versus 31, so it's not that big. That also induces the use of the high performance setup of the EvoSep. That means with a column that is similar to the one we use on the nano-elute. So all in all, it's quite similar. And the results which I have shown earlier with the nano-elute is the one we have obtained while resuspending the samples in a large volume. So that is clearly this difference. I guess you mentioned the difference between the 1,400 protein group ID and the 2,200 roughly. But basically, we reach similar results with the two systems nowadays.
Should show it, huh? The next time.
Yeah.
And the second question I had, at first, you give introduction about TMT and later on about the label-free approaches.
Yeah.
But one thing I'm missing at the label-free is the disadvantage about the time. Yes. In total, I think your gradient takes approximately 30 minutes, and then you can inject the next sample. That means 2 samples per hour. Yeah. If you work with TMT, 18-plex as example, then you have per sample 3 minutes.
Yeah, but that is just comparing, I would say, apple with pears. Let me just try to explain what I mean by this. It's what matters in the end, and correct me if I'm wrong, is basically the analytical outcome. That means it's our view. We shall compare two approaches by the number of, for instance, protein you can quantify in the end. And that's a real difference here. If you mention, you're perfectly right. I mean, half an hour gradient with
outcome, yeah?
half an hour gradient in single cell, you process just one cell. But in half an hour gradient with DIA, you have an analysis depth which is much higher. Not only analysis depth in terms of IDs, but also in terms of data completeness. If you apply that on a 100 cell, for instance, the number of protein you might finally quantify is much higher than if you apply data-dependent analysis, which you are forced to with a TMT approach with the same gradient length. So that's the point. And if you want to compare similar number of protein finally quantified, then when we take it similar, the gradient we can use with label-free approach is much shorter than the one you use with the multiplexing approaches. And that might still evolve with the plex DIA approaches.
Thanks, but I talk only about the runtime, not the analytical outcome, yes? Because
Yeah.
you pointed out different points, and the point is a runtime, yes? And we can point out the runtime per channel, yes? And the total runtime in a DA experiment, yes?
Yes.
Mm-hmm.
And all in all, well, you have to set the cursor between how long you want the runtime and what outcome you want to have. But as said, both approaches have their advantages and drawbacks.
Thank you, everyone. More questions? We are about 15 minutes on delay, but it's actually my fault. It is not yours, Pierre-Olivier. So if there's no more question, we'll move on. Perfect. Thank you. Thank you very much again.